Hi, I'm Ted Lazarus. I'm an internist by training. I focused my clinical career on HIV and AIDS. I've been in clinical diagnostics for 17 years, but I had a 10-year overlap, so I'm not 102 years old. So it's wonderful to be here with you today to participate in a dialogue regarding diagnostics and precision medicine. All of us, including physicians, healthcare workers, and patients, take clinical diagnostics for granted, and honestly, that's a good thing. It means that this valuable tool that provides majority of actionable healthcare information works seamlessly. However, as our knowledge of molecular biology has gone from the study of cells to genes to pathways, and now bioinformatics, so too has there been a fundamental change in what we can and must expect of clinical diagnostics.
Hold on a minute, Ted. Is this not showing up on the screen?
Oh, I'm sorry. I must have knocked it.
Hold on a second. Might have just.
Ah.
There we go. Thanks.
The colors were amazing in those first three slides. You'll just have to trust me. Just amazing. So next generation sequencing is adopting within oncology with the testing of often hundreds of biomarkers at one time. Consortia have formed in the pharmaceutical space to advance the field of personalized healthcare and infectious diseases, often standardizing on current and future generation clinical diagnostics platforms to standardize data output and bioanalysis, thus leading to flexible partnerships between pharmaceutical and diagnostics companies. They're approaching 400 targeted drugs in the pipeline or marketed for oncology. Never before has there been such a diverse number of categories of chemotherapeutic and immuno-oncology agents to choose from. But how to ultimately personalize this therapeutic armamentarium to combine the agents, often in unexpected ways, to maximize treatment is still a dilemma. Here you can see 5 key drivers that are leading personalized healthcare and the transformation of the pharma business. The first is advanced products and personalized healthcare, combined, of course, with their companion in vitro diagnostics and FDA PMA-approved tests. Secondly, platforms globally available, suited for clinical testing. Third, new technologies such as next-generation sequencing, multi-analyte, multiplex testing. Fourth, immune monitoring, longitudinal testing and liquid biopsies. And finally, clinical informatics, actionable insights. So I decided that these topics are so important that there would be no panel discussion or question and answer, and I'll just go through them in a very thorough way. This is something very important. It's something that people don't generally think about. And I just put it up to say that, you know, a test is the diagnostics equivalent of a pill, a tablet, or an injectable drug. The dilemma or the challenge of diagnostics is that you also have to develop the hardware and the software. You don't need hardware and software to take a pill or to give someone an intramuscular injection. However, the tests, what we call the juice in the biz, has to be run on hardware with very sophisticated software, both to make the test happen and to analyze data in real time. It's a very complicated affair. So I'm confident that you know that the diagnostics approval process has become what we call ever more pharma-like in regards to the requirements of very large numbers of patients followed over increasingly longer periods of time. This is especially true for any diagnostic submission related to oncology. However, what is not talked about enough is that the process of reimbursement of these new markers has been fraught with an essential lack of understanding of how these markers will be deployed. Most often, these markers are the gatekeeper for the most efficient use of the drugs that can cost upwards of $100,000 per course of treatment. It's quite amazing what these biomarkers save, both in patient lives and in the cost of care for just a few $100s. And having worked extensively in the area of reimbursement, first with HCFA and now with CMS, I'm often absolutely, as our English friends would say, gobsmacked at how we quibble over pennies, a few dollars, for these extraordinary insights that these tests give us. I always wonder about it. Sally, you brought this up before, and this is a slide that really ultimately shows that there's kind of a ballet, a dance, between getting the drug approved and its companion diagnostic approved. And it's rather tricky, but we make it happen. Ultimately, there's an ecosystem of quality and compliance that must be strictly adhered to, especially as it is applied to the pairing of oncotherapeutics and their companion diagnostics. We see that realized in the FDA-approved drug package insert, where the approved drug is matched with an approved class of test. But there is always a however. And the however is that there is a huge difference between tests of a similar name and specific and total number of genomic targets that are multiplexed in that test. We all need to have a more informed and granular approach to companion diagnostics and realize that they are not interchangeable generics. I think a great example is that EGFR testing has been an extraordinary gatekeeper and has helped us decide which patients will and should be selected for some of the most powerful and effective new drugs. But for the two that are approved, one has double the number of genomic markers, one has half the number. They have different numbers of markers with overall different indications. We've had a very generic approach. It's time to be more mature in the way we look at these markers. When we're able to obtain tissue from a patient, it's important to recognize that the number of diagnostics inquiries that are sought often exhausts the sample at hand. That is why we have developed a multimodal approach wherein what used to require, for example, 5 tests, 5 different hardware platforms, and 5 samples of that tissue can now be done with 1 tissue sample on 1 device. And this is really an extraordinary advance, not just in terms of this very precious resource, which is what Dr. Salat does so many times in terms of inquiries of what's in the freezer to try to learn more for cohorts that have been carefully characterized or for patients who are needing and receiving care. I just want to do 2 quick things very quickly and just talk about liquid biopsy because it's come out. It's a very exciting area. It's euphemistically called liquid biopsy, and it represents technological advances to monitor a tumor non-invasively, specifically and usually through blood testing. We don't often realize that about 20%, for example, of non-small cell lung cancer patients are not accessible for tissue biopsy. In Europe alone, that adds up to about 15,000 non-small cell patients who are currently not tested for their EGFR mutations. So this is a very important alternative in terms of looking at initial diagnosis or recurrence. And liquid biopsy allows you potentially to do something very important. Even when we personalize care, a lot of times that care is delivered in a very standard fashion. And so it's a way to say, have we gotten everything that's there? To monitor the patient afterwards meticulously and to find recurrence at the very earliest time. And finally, liquid biopsy allows a continuum of care from screening, diagnosis, prognosis, stratification, treatment, and monitoring along the entire continuum. I wanted to say a very quick word about NGS. If you look here at what the pain points are, you see that one of them at the very top is reimbursement. Reimbursement is the progression from coding to coverage and ultimately to reimbursement. Can paradigm shifts in care become routinized without reimbursement? Of course not. The first NGS approval was in cystic fibrosis, probably one of the best studied genetic pathways out there. Was better testing needed? Yes, absolutely. Does this approval pave the way for oncology panels running on NGS? I'm not exactly as sure as you are, Sally, but there is something that I am very sure of, and that is that this is a great challenge and a great opportunity. A lot of preparation and honest dialogue with FDA has occurred. It needs to continue. However, we need speed, and so the conversation should and need to be focused on how will we accomplish this instead of who's going to be gaining market share. I think there really needs to inject a little altruism in all of this. Last two slides. So just wanted to mention something about bioinformatics really quickly. Ultimately, bioinformatics is a tool that takes an unimaginable amount of raw data, turns it into a list of variants, and then most importantly, can provide clinical insights that are actionable to help a patient. How do you approve a bioinformatics engine when it's updated every day with insights gleaned from the high-impact peer-reviewed literature? How do you pay for it? I don't know those answers yet. I'm very convinced, though, that we're going to together have those answers and get them with great speed. There are trends already underway to share in an unidentified manner, but a very positive way, and looking for trends for what surprises you. Variants of unknown clinical significance, new trends, et cetera. I think that it's become increasingly important to recognize that this is more important than ever, the era where data must be shared instead of hoarded. And in summary, diagnostics technology is represented by both tests and platforms. The frequency of companion diagnostics testing is going to increase, fueled by the emergence of liquid biopsy for diagnosis and treatment monitoring in cancer and in other chronic diseases. Bioinformatics is to become an integral part for the analysis, interpretation, and reporting of biological and clinical data. Oh, by the way, I just wanted to mention, remember when we were all sold a bill of goods that we'd just be sequenced once in a lifetime and that would be it? Well, that presupposed two things. One, that the sequencing technology would never get better in your lifetime, and therefore there would never be any need to resequence. And two, that the bioinformatics engine that looked at your raw data, that would never get better. So we have a whole paradigm shift now for both public and private payers to re-educate them and say, no, we shouldn't have made those statements. Regulatory review needs to recognize that approved tests within a category can differ widely in their genomic targets, and therefore companion diagnostics needs to evolve into a more sophisticated and granular review process. And finally, optimal utilization of foundational diagnostics is best deployed within the professional dialogue of the caregiving oncologist and his or her pathologist. Proper levels of reimbursement allow future diagnostics to be available locally, and therefore utilization is is monitored through the implementation of best practices at the level of care. Thank you.